good.
It's because the question you asked about this, can we get Chi-C to be in a kind of a stable state? But actually it's not, I don't think it's possible. This is difficult paper, so I will not interrogate on it, but they describe what are the interactions between each of the units of Chi-C, A and B. And then they are all regulating each other, so I don't think getting an equilibrium is not possible. It's too well regulated. Unless you mutate or do something like that, I suppose.
The amplitude of fluctuation decreases by time, so I think the decrease of fluctuation decreases.
Yeah, so of course maybe if you... What happens if you increase temperature to 40-45 degrees?
If it's a chemical reaction, then if it's a chemical reaction, then the period should decrease, but actually, as in the photos, it is decreased, but it's not decreased as according to the Q10.
That's true. That's true. There is a decrease when you increase temperature, but it is not as expected from normal. Right? But of course, if you increase temperature over 40, then I think maybe you could kill the rhythm.
Yes, because it's protein, so it will be.
Yeah, you can a little bit denature or something, right? Yeah. So. That's another for today. This is for today. That's very, very new, so this is why I decided to go on over it together, because it's just April 2017. So this is the last bit of knowledge on bacterial clock. Have you done some work already for Doi Sensei?
It actually is just screening, like looking at the image and choosing the change days that appear at the last number in the region around yes.
I saw. Nice bother. Nice one.
About solvent rest.
Not so interesting. I thought so. It's yeah. I thought that would happen. They are very. Doi Sensei is extremely severe, and he will not give any important work because he is scared that people are going to steal.
I'm just a student, so maybe if I see something very important, I'm not sure that I will understand that it is important.
Yeah, exactly. But he is so afraid that students are going to steal his ideas and talk to other people. Anyway, let's not go.
I understand. So because I think almost every lab has the same thing.
Yeah, like. Last year, there was a guy from agriculture, and he was very interested student, very, very good, but a little bit like you. He wanted to already know how it was developed. He didn't know what to do in the future, and he wanted to know what happens in research laboratory, what's the life. And then I said, okay, please come. And you can follow the seminar if you want to know what we are doing. Come to our seminar. Oh my God. They got really angry because an external people came to the seminar. Come on, it's a student, you know. But, ah well. Yeah, but they are very severe. They are very severe. Conventional, no, like, how do they say? Confidential. Confidential. Students, they want to learn, right? This is why we are here. Did you get the two? Did you get 2 papers?
Yeah, yes. Too much paper.
No, they are very interesting. The nature communication, it's only for your information.
Oh.
It's very, very complex, but I only want... Well, I can give you this 2 minutes. I'll explain to everybody.
Does your laboratory have any research conference or reading conference, like reading textbooks by
Yes.
students?
Every week on Friday, there is a reading.
Oh,
It's like a journal club, it's called.
journal club.
And we are not discussing anything that we are doing in the lab. We are discussing about what people have done from papers.
Oh,
So if you want to come to these kind of things, I could ask the Kyoju if you can come. If you are you coming to this journal club?
What time is it?
It's usually Friday in the morning.
Yes.
Right. But you have lecture, I suppose.
No, I don't have lecture on Friday morning.
Then it might be possible. And usually they send the paper 1 or 2 weeks before, and then students read, and then one student will do the presentation.
So I'm afraid that because I don't have enough knowledge, so that I can keep pace with you.
I don't think you need to worry because many different teams. What I do, the Doi Sensei's team and Yamaguchi Sensei's team, they don't know much. And what they are doing, my students don't know much. So when we do a journal club, usually we try to explain the basics of things. But you can try. You can try once or twice, and then if you like it, you can continue.
is it difficult? More difficult than this class?
Yeah, I think so. I think so. It's probably a little bit more difficult. Because they will show primary paper. They will not show reviews. I try to show you reviews, which simplify things quite a bit. During the journal club, they will give more
research paper.
Yeah. Did I give you a read? This looks the same. This is different types. Okay. Thank you. Okay, so we can start. So there are 2 papers today. The main one, which I think is very interesting, I just saw it today, actually. You can see the date, it's April 2017. So it's really the last things we know about the importance of the circadian clock. Not in mammals, not in eukaryotes, but in bacteria. And I think it's pretty interesting. So we are going to read this one together. The other paper about Kai-C in Nature Communication, you can see this is the same team as Kondo, the paper we had last week. And this one is a little bit complex, but it's on the interactions that each subunit of Kai-C do with its partners. And to have an idea of what happens, you can go to figure 5 directly. You can read it by yourself. We are not going to go over it, but try to read it by yourself. It's very biochemical. And you can see in the figure 5 that not only each subunit of chi-C, so this is the panel A, each chi-C subunit is composed of two domains, the C2 and the C1. And each domain has autophosphorylation activity and autokinase activity, which means it phosphorylates itself or it can dephosphorylate itself. And there is an activity that to do that, it needs to use ATP as well. So there are actually three activities. There is ATPase, there is kinase, and there is autophosphatase. And you can see from the panel A that each pair of subunits sort of regulate each other. This is a little bit complex, but essentially, if you have so maybe let's read the part of the discussion in which we explain this one. I had seen a good part. Where was it? It was an interesting. Page, somewhere page 10. Let me try to find this. Okay. Figure 5, no. Okay. So it's on page 10. It's the first paragraph on the left on page 10. And it refers to the figure 5 that you have on the other side of the paper. So, based on all these observations, we propose the following mechanism for the regulation of chi-C, autokinase, and autophosphatase activities by its phosphorylation state at subunit interface. So they are saying at the interface between two subunits, there are interactions, there are regulatory interactions. The phosphorylation state of a single residue, so this means if you look at figure 5a, they are talking about the two residues S and T. S is serine, and T, this is the yellow thing. S is serine, and T is threonine. So these are the two residues of chi-C that can be phosphorylated, autophosphorylated. You can see where they are? Maybe I should show you this one. Yeah, nice. So what I am talking about is these things here. So what they are writing is that another page. Okay? The phosphorylation state of a single residue affects the ATPase activity and nucleotide binding release within its own reaction unit at C2 domain. So what it is here, S and T, they are two of the sites. Single residue. So this is the serine and this is the threonine of the C2. And the phosphorylation state actually regulates the ATPase activity of that unit. Okay. So not only Chi-C has an auto-phosphorylation, autokinase and autophosphatase activity, but whether these residues are phosphorylated or not will actually promote or inhibit the ATPase activity. Okay? So essentially it means that each subunit regulates each other. This is what actually it means. So when it is phosphorylated, it actually promotes this dephosphorylation. And when it is not phosphorylated, it promotes phosphorylation. So this is actually what it means. But not only there is an interaction at each subunit, but as I told you last time, Chi-C is made of hexamer like this. And each of the monomer has a C2 and a C1 side. So not only each couple actually regulate locally their autokinase and autophosphatase activity, but when they are part of the whole complex, you have very, very, very complex regulation between each of the subunits. So when you have only, so there are two sites that can be phosphorylated, the serine and the threonine. And when you have only one subunit that is only phosphorylated at the threonine residue, this is here, this promotes the phosphorylation of the other subunit. And this is what happens here. So this is not phosphorylated, but one of the subunits, if it becomes phosphorylated, then this promotes the phosphorylation of the other subunit. And then you have a progressive autophosphorylation of Chi-C that then becomes phosphorylated at the P and at the T and the S to become a hyperphosphorylated state. But then when the subunits of Chi-C are phosphorylated at the threonine and the serine residues, they inhibit further phosphorylation. Which means that the only thing that can happen when it's completely phosphorylated is dephosphorylation. And this will continue until Chi-C is fully dephosphorylated, which will bring about a second cycle of phosphorylation, dephosphorylation. And of course, Chi-C doesn't do that by itself. It needs Chi-A and Chi-B to promote or inhibit autophosphorylation and the rate autophosphorylation. But you can see it's a pretty highly regulated system. And I think because of these inter-subunit regulation, pair by pair, and this overall regulation of Chi-C activity, I think it is not possible to actually have a state where if it is normal, if you don't make any mutation or if you don't change the structure of the protein, this is a system that is designed to keep on oscillating for many, many, many days. And as long as you get ATP, these proteins will keep on continuing. And I don't think it is possible to get a state at which they do not oscillate anymore. If you provide all the things that are required to function, I think they will continue forever. But it's true that maybe if you actually change temperature, maybe up to 40 degrees or something, maybe the protein will still be okay. Maybe the protein will not be yet denatured, but maybe the phosphorylation will stop. This is possible. I don't think they have tried 40 degrees, but I think at 40 degrees it might be already not so good for the protein. So anyway, this is the state of knowledge with. This is only maybe 2007, 2013. So this is the last bit on the function of these chi-c. This is only a model. Okay? This is only a model of how it is regulated. And maybe one very nice, I was impressed by the experiment they did. I will show you so that you can read. This will not be part of the exam, okay? It's a little bit too complex, but I was really amazed by the experiment they did here. And this is one of the very clever bits. So they know I have to go opposite. So they have actually made mutations of these chi-c. So they know there are sites that are important for the phosphorylation. So this ST here, these sites are important. And this site here is important for the ATPase activity. So what they have done, they have mutated these sites. They have made chi-c subunits who are mutated. So they have mutated this E, this glutamate site, which is important for ATPase activity. They have mutated it into a different amino acid. And they have mutated the chi-c in which the serine and the threonine were both mutated to alanine. So you cannot have any more phosphorylation. And what they did, they actually mixed wild-type subunits with mutant subunits. And so they have intermediate chi-c who are half made with mutant subunits and half made with real wild-type subunits. And then they can interrogate what is the relationship. If you don't have the ATPase activity, what happens to the phosphorylation of serine and threonine? And if you don't have serine and threonine there, and if you don't have phosphorylation of these serine and threonine, then how is the ATPase activity mediated by the glutamate uridine? So it's very complex, but this is how they sort of tease apart by making chimeric chi-c with mutant subunits. So this is a very elegant experiment. You can read it for your information. It's kind of very elegant experiment. It's all in vitro. They are really using the in vitro system very well there. So it's one of the amazing studies. Okay. So that's enough for this one. And now we are with the review here. And what I like this thing is that not only it's explaining about cyanobacteria, but they also mention about one of the latest discoveries that... Okay, if you think about it, what is our main component? Inside our body, what is the most numerous cell? If you were to count all the cells in our body, what's the main, in terms of number, what is the most numerous cell in our body?
Cells of connective tissues.
That's a good.
Adipose cells.
That's yeah. Some people really have a lot of it. Some people really have a lot of it, but it's a good guess. But it's too limited. Think. What are we made of? What is the cells that contains, or maybe in terms of DNA content? What is the most numerous cell in our body?
Red blood cell.
No.
No.
Yeah, it's very interesting because 10 years ago, nobody was talking about it. But since a few years, this became extremely popular. It's extremely a popular field of research now. And you have people in the United States who are doing all sorts of experiments. Very, very interesting. Sometimes it's very disgusting. But it's amazing. So try to think outside the box. Do you know this expression? Think outside the box. Don't be limited by what you think you are made of. Try to think. Is there not something that we have overlooked? in my lecture I talk to you about eukaryotic cells, and then the liver cells, and then the brain cells, and then the adipose tissue. But isn't there something else in our body? Yeah, see, it's really nice because very few people actually.
Osteocyte.
Huh?
Osteocyte.
Osteocyte. It's a good guess, but that's not correct. So there is a new notion of fact that we are a meta-organism. Meta-organisms mean something more than just human. And mice are the same. It's not just human. We are meta-organisms.
Neurons?
Huh?
Neuron cells? Neurons?
Neurons, no, good guess, but not correct.
Bacterial cells.
Huh?
Bacterial cells.
Yes, we are made more of bacteria than of human cells. And instead of genomic content, we actually have more bacterial genome than human genome. But of course, it changes very quickly, and it depends on your food. But now what researchers are trying to understand is, what is the relationship between our own cell and between bacterial cells? And they are doing what is called the microbiome, and they have discovered very interesting things. Now, this is not part of this lecture, but I went to Hawaii at a conference on these synthetic biology, and then they have discovered very interesting things, that you can cure some disease by simply injecting some type of bacteria in the gut. They are really kind of interesting interactions between our body and bacterial cells. And this review kind of mentioned that. So this is very interesting. So we are going to see it together. And I think it's nice to kind of read together as before. So maybe let's start with the first paragraph. Please, Gwen-san. Don't need to read the abstract. The first paragraph is okay.
Many bacteria experience substantial environmental changes during the day and night. Most free-living bacteria are exposed to daily cycles of light and darkness and/or temperature. The gut microbiota is also exposed to daily changes in the intestinal environment. As most animal hosts eat on a daily schedule, usually during the day for diurnal animals and during the night for nocturnal animals, which creates a daily rhythm for feast and fast in the digestive system. Although endogenous circadian rhythm were once thought to be an exclusive characteristic of eukaryotic organisms, all defining properties of circadian clocks were also found in cyanobacteria. Many processes are circadian in cyanobacteria. For example, nitrogen fixing unicellular cyanobacteria rhythmically fix nitrogen during the night, approximately 12 hours out of phase from photosynthesis, which peak at midday. This adaptive biochemistry, as nitrogenase, a nitrogen-fixing enzyme in these cyanobacteria, is inactivated by even tiny amounts of oxygen, and its activity is thus incompatible with photosynthesis, which releases oxygen. This is one simple example of why bacteria may have evolved timekeeping systems to coordinate metabolic events during the day to optimize performance and/or to temporarily separate potential conflicting metabolic processes. Whereas the timekeeper of cyanobacteria is well established as a prototypical circadian clock, other bacteria may have evolved alternative timekeeping systems, such as an hourglass timer, which confers a fitness advantage. Okay, thank you very much. So be careful, just properties, not properties. Properties is fine, it's easier to say. Any question, I think it should be okay. It's very general explanation. So the first time we see, I have not explained to you yet what is an hourglass, but when people were without much evidence
from biochemical studies in mammals or bacteria. They were trying to understand what is the clock that is in our body. Is it really like a clock that oscillates, or is it an hourglass? Do you understand the difference between an hourglass and a clock? Of course, they say hourglass timer is a simple non-oscillatory timer that is set up in motion and keeps track of time linearly and does not self-sustain a cycle. So yeah, this is okay, but it doesn't really maybe provide much of an explanation. So this is exactly an hourglass timer. It's the difference between your watch and this little Snadoke that you use for eggs. This hourglass timer, the Snadoke, the sand clock, it's something that actually measures a duration of something. You start the sand clock and it will measure 3 minutes or 20 hours. So it will measure the time elapsed. And once the time has elapsed, then something must come and start it again. A clock is not like that. A clock will keep on oscillating. It doesn't need to be started because it's always there and it's always keeps track of time. And there were people that believe in the beginning that maybe the clock in mammals is like an hourglass timer. Maybe every day, because the sun rises, our clock starts ticking, and then it will run out at the end of the night and needs to be started again with the light. And if you remember, I gave you an experiment earlier about cells. You have cells in culture, and the people, there were people from Switzerland, they were measuring clock gene expression from these cells. And they found out that it is only when you shock the cells with serum that the cell start oscillating. And some people say, this is because it's kind of an hourglass clock. It is starting once you give a dexamethasone or a serum shock. But it's not really it is not self-oscillatory. So this is they were discussing. And then they found out that it is really oscillatory, and it's only the rhythm can only be seen after a shock because all the cells become synchronized. So this is why they noticed it was oscillatory, not an hourglass timer. That being said, there are some hourglass timers that exist in living things. Okay, not everything is a self-sustained oscillatory clock. Okay. Any questions on this first paragraph? That should be okay.
What does this serum shock mean?
Serum shock. Sorry. Serum shock. Maybe just like this one. Serum shock. Do you remember this serum shock? Do you remember this? This is Koyo. Do you remember serum shock? You remember this?
The experiment.
Yeah, serum shock. Do you remember serum shock? No?
I don't.
Do you remember serum shock?
No.
Do you remember serum shock?
There's the mesosome experiment like this.
Yeah, so serum shock or dexamethasone, this is two different kind of shock. Dexamethasone, it's a chemical, it's a corticosteroid. It's a glucocorticoid. It will activate the glucocorticoid receptor. And serum shock contains many things. Okay, it contains all sorts of hormones. And normally, if you cultivate cells, this is time here, and here this is clock gene expression. So maybe per 1, for example. And if you measure per 1 expression in cell culture, you actually don't notice any rhythm. It's just completely flat. So the conclusion of that is that cells do not have clock. However, some clever people have used these shock experiments, and they found out that when you give, let's say for 2 hours, for only 2 hours, you put into the cell culture medium a high amount of serum. Normally, medium only contains 5 or 10% serum. But for 2 hours, maybe you put 50% of serum. Or you add dexamethasone. For 2 hours, and then you wash. Then what you get, after this shock, then you have very nice oscillatory gene expression. So people were saying maybe that's what's happening, but this gene expression will actually kind of go up. And if you give again 2 hours of serum shock, when the rhythm disappears, then it will start again. So some people were saying maybe this is like it's like a sand clock. So you need to start the clock, but the clock is not self-sustained. But actually what it means is simply all the cells here, they are still oscillating, but every cell is out of phase with each other. So actually when you measure the whole population of cells, you get an average of all cells. And when you give the shock, they all become synchronized, and then when you measure gene expression from the whole population, then you can measure. So this is what happens when they speak about serum shock. Rajulu, you understand?
Yes.
Any other question? Okay, then let's go to the next paragraph.
What might other functions of a daily timekeeper in bacteria be? The most common hypothesis is that having about 24-hour timekeeper enables the cell to anticipate and or prepare for the daily transformation of environmental conditions that affect many bacteria. Anticipation might be most easily discerned in the case of photoautotropic bacteria, such as cyanobacteria, owing to the fitness disadvantage of maintaining energetically expensive photosynthetic pathways during the night. Thus, bacteria that anticipate dawn by activating quiescent photosynthetic processes late in the night could have an advantage over bacteria that merely react to dark to light transitions. Anticipating daily changes might also facilitate protective responses in bacteria that are exposed to the damaging effects of sunlight, especially UV light. Even visible sunlight can be absorbed by cytochromes in the electron transport chain and affect metabolism. The effects of sunlight inspired the escape from light hypothesis, which proposes that circadian clocks evolved owing to the selective pressure of daily cycles of light and darkness, in which light impaired growth.
There are maybe some concepts here, which is kind of interesting. Anything that on which you would like to have more explanation? 大丈夫ですか? What about electron transport chain? Cytochrome in the electron transport chain. What are we speaking about there?
Photosynthesis pathway.
Actually, I don't think so. I don't think so. I don't think they are speaking about photosynthesis there. Well, they are talking about photosynthesis, but they are talking about damaging effect of light, right? So light doesn't have damaging effect on photosynthesis. Photosynthesis need light to function. But there are other parts of metabolism which are affected badly by light, like the electron transport chain and cytochrome. What are we speaking about here? Actually, this also exists in mitochondria. This electron transport chain. What is photosynthesis? What is the main product of photosynthesis? Glucose, yes. Photosynthesis is the synthesis of sugar from carbon dioxide. But the bacteria need something else. As you do, you get glucose from your food, you cannot make glucose. You can make glucose from glycogen in your liver, but you cannot make new glucose. Well, you can use amino acid to make glucose, but you cannot fix carbon dioxide to make glucose. Plants can do. But bacteria need something else. They use glucose for what? By photosynthesis, they make glucose. And this allows them to do what? This is the other part of the metabolism of bacteria and plants as well. It's the same why you need glucose. Why do you need glucose? Producing ATP. So cyanobacteria, just like plants, they are using photosynthesis to make glucose, and when it's dark, they are using glucose to make ATP. And this electron transport chain is it's a machine, it's a big molecular machine that actually make ATP from glucose. And from other things, it also needs electrons. This is why it's called electron transport chain. Electrons, notably from ADP, NADPH and NADH. So this is called the electron transport chain, and it's something that makes ATP using electrons. But the problem is that these proteins that make ATP by exchanging electrons, they are very sensitive to light. And if they get light, they actually don't function very well. So this is what it means. This is what it means.
No.
What do you mean, no?
The last part you said
What do you mean? So what I mean is, you can essentially say there are two phases. They are mostly talking about the relationship between light and darkness, right? So, during the light phase and the dark phase. If you are a bacteria, when there is light, you will do photosynthesis. Photosynthesis. Because you've got light and you can receive light, okay? But during the dark phase, you will actually make ATP. But while you make ATP, if you have light, it's not so good, because the efficiency of making ATP will decrease. Because light will disrupt all these proteins that are synthesizing ATP. So it is not good to have light when you make ATP. This is essentially what they say. Okay? This is essentially what they say in the text. So they say, even visible sunlight, so not only UV, but normal sunlight, can be absorbed by cytochrome, which is the name of the proteins that make ATP. In the electron transport chain, this is the assemble. This is the group of proteins of cytochromes that make ATP. Even visible sunlight can be absorbed by cytochromes in the electron transport chain and affect metabolism. So the effect of sunlight inspired the escape from light hypothesis, which proposed that circadian clock evolved owing to the selective pressure of daily cycles of light and darkness, in which light impairs growth. So you need light to make photosynthesis, but there are certain things, there are certain reactions you want to make for which light is not good. So sometimes when you want to make these things, you have to escape from light. And for that, having a biological clock that tells you that if you have a clock that tells you before night falls that, okay, light is going to disappear, get ready to make ATP. You can already prepare all the machinery to make ATP before even night comes. And then at the end of the night, if you get light and you are surprised because you have light, you are still making ATP, but suddenly light comes and it makes a lot of damage to your system, this is not good. But if you have a clock, then the clock will be able to tell you, okay, be careful because light will come soon, so you should shut off ATP synthesis and be ready for when the light comes. So it allows you to anticipate when light comes and escape the effect of light. So this is what they are discussing. Okay, is that clearer?
Yes.
Okay. So let's go for the next paragraph.
A gut bacterium that anticipates when fresh nutrients are entering the digestive tract have an advantage in the competition for resources. Furthermore, gut bacteria may also benefit from daily timing when they attempt to survive outside of the host following excretion, in a similar manner to bacteria that are always exposed to the external world. Therefore, bacteria that have a daily timekeeping mechanism, even if it is not a complete circadian system such as those found in cyanobacteria, probably have adaptive advantages. In this review, we highlight recent mechanistic insights into circadian oscillators in cyanobacteria and timekeepers in other bacteria. Moreover, we consider the potential adaptive value of those oscillating clocks and hourglass timers. We also examine recent research on the evolution of proto-circadian rhythms and how the relationship between a host and its microbiota is influenced by biological timekeeping.
How about this one? That should be okay. Microbiota, this is just the, the population of bacteria that we have in our system. Okay, so I will continue and read the next one. Timekeeping mechanisms in cyanobacteria. In cyanobacteria, the clock system controls multiple output processes, including the transcriptome, the proteome, the timing of cell division, nitrogen fixation, chromosomal topology, and metabolism. In photoautotrophic cyanobacteria, there are changes in metabolite composition during the day and night, and, owing to self-sustained function of the clock, these changes persist in continuous light. Even the redox status of Synechococcus elongatus cells oscillates in continuous light and may influence the oxidation status of peroxyredoxins. Interestingly, changes in metabolism that depend on the light and dark cycle, in particular redox status and/or energy levels, can directly entrain the cyanobacterial clockwork. Metabolic feedback might even provide mechanisms to compensate for temperature and other changes to enable the clock to maintain accurate timekeeping. What does redox mean?
Reduction, oxidation.
Yeah, reduction, oxidation. And peroxyredoxin, remember we have seen that last week. Okay, then let's go. Should be okay. Unless it's okay, please stop me. Otherwise, we are going to continue.
The core mechanism of the circadian clock in eukaryotic cells is widely thought to be based on the transcription-translation feedback loop. Although there is evidence that this model may be incomplete or inaccurate. In cyanobacteria, initial experiments also support a transcription-translation feedback loop model. However, the remarkable discovery that three cyanobacterial clocks proteins, Chi-A, Chi-B, and Chi-C, could reconstitute a circadian oscillator in vitro for many days led to our current understanding of the clock system in cyanobacteria. A biochemical oscillator is the core pacemaker, which operates in and regulates a larger transcription-translation feedback loop that in turn controls outputs and replenishes the essential proteins of the oscillator. At the present time, the cyanobacterial clock is the best understood of any circadian mechanism from a biochemical and biophysical perspective.
Okay, thank you very much. And that is very true. All this this relationship between Chi-C, Chi-A, Chi-B, it has been really precisely investigated. Much better than all these interactions that exist between clock, bimal, per, because there are so many of them, right? It should be also okay this time, right? Okay, let's continue.
The key component of the cyanobacterial clock is Chi-C. This protein has an astonishing range of biochemical activities, including autokinase, autophosphatase, phosphotransferase, and ATPase, and is the core of rhythmically associating and dissociating nanocomplex. Chi-C is a double hexamer that consists of two similarly sized rings, C1 and C2, and a carboxy terminal region in C2 that interacts with Chi-A through protruding peptide tentacles. Both rings catalyze the hydrolysis of ATP. As described below, the ATPase activity of the C1 ring provides the rate-limiting reaction of the timing circuit, whereas the ATP that is hydrolyzed by the C2 ring catalyzes sequential phosphorylation and dephosphorylation reactions of serine and threonine residues. The level of Chi-C phosphorylation is a convenient marker of circadian phase in vivo and in vitro, including when transcription and/or translation are suppressed. As neither transcription nor translation is required for Chi-C phosphorylation to oscillate, it is a post-translational oscillator.
Okay. And then we can continue directly to the next one, which will provide a bit more explanation.
Phosphorylation and the nanocomplex cycle. The status of Chi-C phosphorylation is not merely a marker of the circadian phase. It also regulates the association and deassociation of the Chi-A, Chi-B, Chi-C nanocomplex, thereby switching Chi-C between its autokinase and autophosphatase states. The rhythm begins with an unphosphorylated Chi-C. Chi-A stimulates the intrinsic autokinase activity of Chi-C through interaction with the C2 terminal tentacles. This leads to the phosphorylation of two key residues in the C2 domain, T432 followed by C431. Dephosphorylation proceeds in the same order. Phosphorylated threonine 432 followed by phosphorylated serine 431, such that the overall process from the hypophosphorylated to the hyperphosphorylated and back to the hypophosphorylated state of Chi-C follows the sequence.
Yes, you can skip this one.
The dephosphorylation phase proceeds, at least partially, by regenerating ATP from C2-bound ADP and the phosphates that are bound to threonine 432 and serine 431. This remarkable phosphotransferase activity limits the overall consumption of ATP, which possibly preserves clock function when cellular ATP concentrations fluctuate. The cyanobacterial clock accurately tracks time during extended exposure to the dark, when cells are not able to produce ATP by photosynthesis.
Okay, thank you very much. So I would say one very important thing here is to note this is the last paragraph that I told you, and we saw before, that Chi-C has an intrinsic ATPase activity, so it can synthesize its own ATP. It cannot make all the ATP it needs, because if you don't provide the ATP to the mix of Chi-C, Chi-A and Chi-B, it will not oscillate for long. You must provide ATP, but still, it doesn't need much ATP to function, because it can regenerate the ATP that it's using to a certain extent. So that's very important for something that is self-sustained. And you can see that the oscillations of Chi-C is really a dephosphorylation followed by it's a phosphorylation followed by a dephosphorylation, progressive from one subunit to the other. And there is as well some exchange of subunits that can happen between these double hexamers. So it's extremely complex. But we have already seen this one. So unless there are questions, I'm going to read the next page. Is there any questions? It's okay. So I'm going to read the next part on 2234. Concomitant with the phosphorylation and dephosphorylation cycle is a rhythm of association and dissociation of the Chi-A, B, C nanocomplex. At the beginning of the nanocomplex cycle, Chi-A rapidly interacts intermittently with the C2 tentacles of Chi-C. This transient interaction stimulates the phosphorylation of Chi-C or inhibits dephosphorylation, thereby shifting the reaction towards phosphorylation. And then S431 of Chi-C achieves the T432 PS431 state in the phosphorylation sequence. Chi-B binds to Chi-C. A recent study reports that Chi-B exists in two diametrically different conformations. A ground state Chi-B that does not interact readily with Chi-C, and a substantially fold-shifted state, fold-shifted Chi-B, that binds to the C1 domain. The FS Chi-B state is conformationally similar to a structural motif of SAS-A, which is a histidine kinase that interacts with Chi-C and transmits its oscillatory information. FS Chi-B competes with SAS-A for binding to Chi-C and thus regulates the downstream transcriptional cascade of SAS-A and its response regulator RPA-A. In addition to displacing SAS-A from C1 of Chi-C, Chi B forms a complex with C1 domain that also establishes sequesters and inactivates Chi A. So that Chi A can no longer stimulate the phosphorylation of Chi C, and Chi C switches to its autophosphatase mode. When Chi C fully dephosphorylates, Chi B and Chi A are released, which restarts the nanocomplex and phosphorylation cycle. So that's a very complex paragraph about what actually is the interaction between Chi A, B, C, but we also see SAS-A and RPA. So can you just briefly tell me what is really the main role of SAS-A and RPA based on figure 2? So you don't need to understand all these relationships, too much detail, okay? But you have the Chi A, Chi B, Chi C complex, and you have this cycle of phosphorylation, autophosphorylation. But what is the role of SAS-A and RPA? What does it provide?
Stimulate the transcription of Chi.
Yeah, so not only that, but it stimulates the rhythmical transcription of many output genes. So the Chi A, B, C is the oscillator in cyanobacteria, but it sort of gives timing information to output genes who become rhythmically transcribed, just like in mammals. And SAS-A and RPA, they are the proteins for that. They interact with Chi A, Chi B, Chi C, and they can feed information forward to promoters, but they can also enhance the cycle of Chi A, Chi B, Chi C by promoting rhythmic expression of the Chi A, B, C cluster.
When Chi A bind to Chi C, then the Chi C are when Chi A bind to Chi C, Chi C are phosphorylated at both places. Serine for 431 and 432.
So you can take a look. You can take a look at figure 3. This is a nice little picture there. So in the beginning, the double hexamer Chi C, so this big purple thing there, it's not phosphorylated. You have C2 and C1, and it's not phosphorylated. And the ATPase activity is not very high. The ATPase activity, this is the graph that you have, the yellow graph. And then you have GS Chi B and FS Chi B as well. But then what happens is that you have association of Chi A, which triggers the beginning of phosphorylation of Chi C. Okay? And then because of that, the ATPase activity increases. And the cycle proceeds. Chi C becomes hyperphosphorylated. And then you have another factor, is that the FS Chi B starts to bind to Chi C. And the rhythm progress where you have inhibition of ATP hydrolysis, Chi C switches to autophosphatase activity because it became hyperphosphorylated, so it switches to autophosphatase activity. And so Chi C becomes dephosphorylated. Chi A is sequestered to a different part of Chi C. And then further dephosphorylation continues. Chi A and Chi B dissociate, and it's back to the original state. So I would say the first event is Chi A interact with Chi C, and phosphorylation starts. So yes, Chi-A rapidly interacts intermittently with the C2 tentacles of Chi-C. This transient interaction stimulates the phosphorylation of Chi-C. So this is really the first thing that starts the phosphorylation cycle. This is why you need Chi-A. If you don't have Chi-A, then Chi-C will not, the phosphorylation of Chi-C will not happen. The cycle will not happen. Okay? Right. Let's continue. The slow pace of ATPase activity. It will be, no, Guillaume, right? Because I read the last one, right?
The slow pace of ATPase activity. How can this biochemical oscillator maintain such a long time constant? For example, a period of 24 hours. Are the phosphorylation and nanocomplex cycles the core pacemaker of the cyanobacterial circadian clock? Similar to the transcription translation feedback loop, there is nothing intrinsic about these cycles that makes them oscillate with a 24-hour period. Certainly, phosphorylation events and protein associations and dissociations can occur very rapidly. These biochemical reactions do not dictate a slow 24-hour timekeeping. Kondo and co-workers have championed the hypothesis that the ATPase activity of Chi-C is the core rate-limiting reaction. This reaction is very slow. Only 15 to 16 ATP molecules are hydrolyzed by one Chi-C monomer per day and is temperature compensated. Moreover, an analysis of Chi-C mutants in vivo and visceral show a strict correlation between the ATPase activity of Chi-C and circadian frequency, which implies that the circadian period depends directly on the energy that is provided by ATP hydrolysis. There is even evidence for a highly damped oscillation of the ATPase activity of Chi-C in the absence of Chi-A and Chi-B.
Okay, thank you very much. So this we kind of briefly mentioned last week, that the reaction, the ATPase reaction of Chi-C is very, very slow. Much slower than most of other proteins that mediate this kind of reactions. And it's very important because it has to tick with a period of 24 hours. So it has to be slow. It cannot be quick, because if it's quick, it can be finished within minutes. Okay, so let's continue directly. Unless you can stop if we have questions. Okay, please continue.
However, is the ATPase hypothesis as the core pacemaker just an infinite regress with another process underlying the slow rate? Given that many ATP hydrolyzing reactions have very fast rates, can we explain the sedate tempo of the Chi-C ATPase? A structural study identified features in the C1 domain of Chi-C that might explain its slow ATPase activity. The lytic water molecule that is involved in nucleophilic attack on the gamma phosphate of ATP is positioned away from the optimal position, so that hydrolysis is less favorable than in the catalytic sites of more typical ATPases, such as kinesin and F1 ATPase. A second source of the slow activity is slow cis-trans isomerization of a crucial peptide bond, which increases the activation energy barrier of the ATPase reaction. These two features decrease the probability of ATP hydrolysis and thereby establish a remarkably slow but stable ATPase cycle in the C1 domains of the Chi-C hexamer. This landmark study provided the first biochemical and biophysical explanations of how a circadian oscillator could have such a long time constant.
Okay, thank you very much. So I guess maybe based on the previous lecture that some of you got, you should be able to understand this paragraph about the activation energy barrier. So it's all about the shape of the Chi-C protein that makes that all the positions of atoms that are involved in this hydrolysis, which is ATP hydrolysis, is actually not well positioned compared to others. It takes more energy to do it. And this activation energy, do you understand what is activation energy? A second source of the slow activity is slow cis-transisomerization. So it means there is the configuration of the atoms changes in the Chi-C. Okay? Isomeric means the same formula but a different conformation of the atoms relative to each other. And this increases the activation energy barrier of the ATP reaction. What is the activation energy barrier?
Amount of energy needed to start the reaction.
The minimum, the minimum energy needed to start a reaction. So all reactions proceeds like this. Even if it is a spontaneous reaction, then the free energy, so let's say this is the free energy, you can say E, it's easier. Let's say this is the free energy, or delta G, or whatever. Then the energy of the products will be lower than the energy of the reactants. Okay? So even if the reaction is spontaneous, it will not happen very often quickly. Why? Because this molecule has a certain amount of energy, and this molecule is lower. So you could say, okay, so it's going to happen very quickly, spontaneously, but no, because in all chemical reactions, if you actually look at the energy, it goes like this. These little bumps here is called the activation energy. So some of these molecules will sort of shock other molecules, and there will be random distribution of energy. They will move around, they will twist. Some may be bombarded by other atoms or electrons or whatever. And some of these molecules suddenly will have just enough energy to be able to jump this thing here. And usually what enzymes do, so for example, let's say this is a dimer of glucose, and this is monomer, so this is a very spontaneous reaction. And if you have an enzyme that mediates this reaction and breaks up the dimer of glucose into its monomer, then what the enzyme will do is that the enzyme will actually decrease the activation energy, so that more molecules will be able to be transformed. But what they say is that actually what Chi-C does, is actually it increases the activation energy. So all enzymes so far decrease the activation energy. But actually what Chi-C does is that it increases the activation energy. So this limits the likelihood of ATP going into the reaction. So this is opposite. It's a kind of an enzyme, but a very specific enzyme, because a very strange enzyme, because it increases the activation energy. So it makes the reaction less likely. And so this is why it takes so long for it to happen, instead of very, very brief, as usually happens with ATPase. Okay?
Why they say that Therefore, Chi-C monomer exchange and Chi-A sequestration ensure that Chi-C hexamer remains synchronized in both translation. The last question, the last sentence. The last sentence.
Sorry. 235. Last question.
The last sentence, I don't know because
this landmark study provided.
No.
Sorry.
Therefore, Chi-C monomer exchange and Chi-A. Because I'm not.
What did I just read? What was the last? Sorry.
At the end of 234 page.
At the end of 233.
226?
Hey, 26?
What?
Yes,
I
I'm
thought
sorry.
you were this landmark study provided that.
Yeah, this is what I read.
Oh, I'm sorry.
This is why I was confused. Okay, so we will see that later.
No, I'm sorry because I didn't look at this page, so I was reading this and then I look at this and I don't understand anything.
Allez! No, I was just reading 234, last paragraph of 234, from however to time constant. Right? This is what we just read, right? Okay. So then let's go to 235. And that would be you.
Cycle with cycle. Robustness and synchronize. The aforementioned study reviewed transcription, feedback loop, negative feedback loop, phosphorylation nanocomplex and ATPase cycles in the cyanobacterial blood work. Why there are so many cycles? An analysis of the relationship between the post-translational oscillator and the transcription-translation feedback loop found that they have a definite hierarchical interdependency. The post-translation oscillator is the core pacemaker.
You can say PTO, it's easy.
Whereas the TTFL is a slave oscillator, which names when the PTO stops and is no longer temperature compensated under some metabolic conditions. The TTFL feeds back into the PTO through the synthesis of the nuclear proteins, which can reinforce and or fine-tune the core PTO pacemaker. This coupled oscillator system is more resilient to intracellular noise. Moreover, an analysis of cells in a microfluidic device found that the clocks of individual cells were less stable and synchronized was not maintained among cells in the population in the absence of TTFL. The advantage of coupled oscillators cannot only be demonstrated mathematically, it also makes sense biologically. The TTFL controls transcription and translation, whereas the clock-specific nanocomplex, which is composed of Chi-A, Chi-B, and Chi-C, is a mass-action biochemical oscillator. These two types of oscillator are likely to be sensitive to different kinds of intrinsic and extrinsic cellular noise. For example, cell division, transcriptional noise, fluctuating metabolites, and so on. Moreover, it is very likely that the core ATPase reaction is stabilized and temperature compensated through its interaction with the phosphorylation nanocomplex and TTFL cycles. Therefore, the three nested and interacting cycles, that is the TTFL phosphorylation nanocomplex and the ATPase cycles, provide a resilient circadian system.
Okay, thank you very much. So, one thing I don't understand is they say a mass action biochemical oscillator. Can any of you explain to me what is a mass action biochemical oscillator? If not, then I will get some information and explain next week. A mass action biochemical oscillator, that is interesting. So let's look at that next. I will provide more explanation about this one next week. Microfluidic device. Do you understand? Microfluidic device. it's also one of the very popular tools now to analyze stuff. It's it's a device that contains very, very, very small chambers and channels with which you can analyze single cells and see what happens within single cell. And it's called microfluidic because it's very, very small. It's sometimes the size of a computer chip. And then inside you have very small channels in which you can actually make things circulate. So you can have an input here and an output. So some of them are very complex. But usually they use these things to analyze single units of single cells or single protein or whatever. And it's called microfluidic because they usually a fluid that is pumped into these systems. But the design of it is very different. Everybody has its own design. So mass action, I'll give you more detail later of what exactly do they mean by mass action biochemical oscillator. Okay, some questions? Resilient, you understand? Resilient. Resilient means resistant to changes, right? Okay, then let's continue. A separate but related issue.
A separate but related issue regarding robustness is synchrony among the Chi-C hexamers in the PTO. Remarkably, in vitro, Chi-A, Chi-B, Chi-C reactions can oscillate for up to 10 days without detectable dampening. Why is the period of each Chi-C hexamer not sufficiently different from every other hexamer in the reaction so that the phasing in the population of hexamers becomes asynchronous after a few cycles? There must be a mechanism or mechanisms to keep the hexamers synchronized. Indeed, at least two such mechanisms are known. First, a Chi-C hexamer can exchange monomeric subunits with neighboring hexamers, and this monomer exchange maintains synchrony of the phosphorylation status among the Chi-C hexamers in the population. Another important factor for maintaining synchrony among Chi-C hexamers is the sequestration and inactivation of Chi-A activity during the dephosphorylation phase. Therefore, Chi-C monomer exchange and Chi-A sequestration ensure that Chi-C hexamers remain synchronized in the PTO.
Thank you very much. I think this is really fascinating. So that's true. These people, this Kondo has shown that Chi-C can oscillate in vitro for 10 days, but there are many Chi-C copies, right? It doesn't have only one protein, it's a mix of protein. Why are they keeping in phase so well in vitro? And then they actually provide some of the mechanisms is that Chi-C can actually exchange their subunit so that they are always in phase. So it's a self-synchronization mechanism. It's really fascinating. Sequestration, Wakaru. Do you understand sequestration? Sequestration is to put it apart, to keep it protected from other things. This is what sequestration means. And what they mean is written in the figure 3. And in the figure 3, you have in the beginning Chi-A is on top, right, associated with the tentacles of Chi-C. And after some time, when Chi-C is hyperphosphorylated, then you can see the blue protein, which is Chi-A, they go somewhere else on the bottom of Chi-C. So this is a sequestration. They are put aside on a different place where they are prevented to react and to further phosphorylate Chi-C. This is what sequestration means.
Where the kind A, where did they store the Chi A in to make them apart from Chi-C?
This is a very good question. I don't know. How the sequestration happens, I don't know. So apparently they are using a different, on the model here, they are using a different domain of Chi-C to sequester. So presumably Chi-A is still associated with Chi-C, but in a different location. And maybe depend on Fs Chi-B as well, it seems to be. But exactly how it is sequestered and how do they prevent Chi-C to Chi-A to react back with Chi-C, I do not know. I don't think, this is still a model, eh? This is still a model. But apparently, only when dephosphorylation of Chi-C proceed can the sequestration of Chi-A stops, as you can see on the figure 3. It's only when dephosphorylation occurs that Chi-A and Chi-B can dissociate and start again. But the mechanism of sequestration is not known. Is it the tentacles that bring Chi-A on the other? We don't know. Okay. So do they cite anything? Do they cite any I will see what has been discovered in terms of sequestration, but I don't think that part is very well explained. But I will check what's on the publication. Okay, let's maybe continue. We have 10 minutes, so let's read the first paragraph of this next section.
The sun also rises for other bacteria. Cyanobacteria, all three Chi proteins are required for circadian oscillations in vivo and in vitro. Homologs of Chi-B and Chi-C are distributed widely among both photosynthetic and non-photosynthetic members of the eubacteria and the archaea. But Chi-A is found only in cyanobacteria. Even among cyanobacteria, the Chi-A gene was lost during evolution in the important cyanobacterial genus Prochlorococcus, which retain Chi-B and Chi-C and has daily rhythms, but not a sustained circadian rhythm under constant conditions. The loss of the Chi-A gene from Prochlorococcus may have de-evolved an original Chi-A, Chi-B, Chi-C circadian clock to a simpler timer that is based on only Chi-B, Chi-C and keeps track of time similarly to an hourglass. Another example of a potential hourglass timer is found in the halophilic archaeogenus Halobacterium, which has a single domain Chi-C, but neither Chi-A nor Chi-B. Halobacterium exhibit light, dark, and train daily transcription, but not sustained oscillations under constant conditions. Does this mean that all three Chi proteins are required for self-sustained circadian oscillations, or is it possible that a circadian clock could be composed of Chi-B and Chi-C, or even just Chi-C alone?
Okay, thank you very much. And on this last question, we are going to adjourn the meeting. So I will look for more information on the mass action complex and on the sequestration mechanisms. That's true, it's a good question. How does it happen? How can Chi-A being associated with the tentacles suddenly change position? No. How can it be sequestered? They don't explain this one in this paper, so I'll look for more information and tell you that next week. Don't forget, next week we are having midterm test. It's going to be an interview. Okay, it's going to be an interview. Maybe 30 minutes preparation of question, and then a small 5-10 minutes interview on what you have written. Okay. It's going to count as well. Well, we'll see. If you are doing well, it's going to count. If you are not doing well, it's not going to count. So you don't have anything to lose for this.
So, so do you need to memorize, for example, the mechanisms, like in the first paper we read?
No, I mean, all single thing, like what protein interacts with, there was a huge amount of protein and a huge amount of histone methylation site and which lysine of which histone, and this is not needed. General mechanism is enough. If I ask you, what is the biological clock in mammals? How does it function? What are the mechanisms that support its function? Okay. Of course, some of the names I will expect of you to know. Like, for example, if you should know a few names, what would it be for the biological clock in mammals? In our cells, what would be the name that you should at least remember? Huh? Yeah, the core protein, BMAL1, clock, PER1, PER2, cry. These things, the basic oscillator, and know that they can interact with each other and that they regulate many things, like, for example, chromatin modeling. But exactly what they interact with is not that important. The basic mechanism is more important. What does it do? How does it regulate? Okay. This is what is important. So there will be, maybe, we are not so many, so I think each of you will have 2 questions. At random, you can pick 2 questions from a bag. This is what usually I do. And then you prepare your questions and then have an interview. Okay. So that means if it's a real rehearsal of the final test, it means that each of you will need to come 10 minutes later than each other. So because I want you to have exactly 30 minutes of preparation. Otherwise it's not fair. The last person to pass the interview will actually prepare for 50 minutes. Right? Because 10 minutes of interview for each. So let's say, if it's alright, you are going to come at 4:30. You are going to come at 4:40. And maybe you will be already there because you have the previous lecture. So come at 4:30 because he follows the next. Then you come at 4:40 and you come at 4:50. Let's do that. So you start preparing at 4:30. You start preparing at 4:40. You start preparing at 4:50. Okay, so let's do that. And she is not there, but she will come whenever she comes. Okay. All right. So if you have any questions, please ask. Otherwise, it's the end of the meeting. Thank you very much.
Prepare without paper.
No, I give you a paper so that you can write.
Review paper.
Yes, without.